Angus Australia has observed exponential growth in the number of genotypes submitted for genetic evaluation purposes in recent years, as this graphic shows. With over 300,000 genomic profiles now available, it is important to continually review how to optimally utilise the genomic information within the evaluation with the aim to providing the highest accuracy estimated breeding values. The following two enhancements, as presented by Dr. Angus Swan from the Animal Genetics and Breeding Unit, focuses on genomics area, specifically an increase in genomic relationship weighting and incorporation of genomics in docility.
Hello everyone, I'm Angus Swan, one of the team behind the development of breed plant systems used in the TransTasman Angus Cattle Evaluation. In this video we'll be looking at an enhancement to the evaluation to make better use of genomic information. Genomics has revolutionised genetic evaluation systems over the past decade, with Angus Australia being a leader in adopting single-step analysis procedures, combining phenotypic data with pedigree and genotypes. With large reference populations for a number of key traits and around 300,000 animals currently genotyped, the TACE system has become a very powerful single-step analysis. One of the key features of single-step is that it can more accurately track shared DNA through genomic relationships between animals. In the pre-genomics era, relationships between animals were tracked through the pedigree, and all animals of a certain relationship type were assumed to share the same proportion of DNA. In the example shown here, all of these progeny have an assumed relationship of 0.25 with their grandsire through the pedigree, or 25% shared DNA. With the benefit of genomic information, however, we can get a much more accurate picture, and in this stylised example, the genomic relationships vary from 0.23 to 0.27. The single-step method allows us to balance pedigree and genomic relationships according to how confident we are in their relative accuracy. When single-step was first introduced, there was a degree of uncertainty about genomic testing, and an equal weighting of 50% was deemed appropriate. But as genotyping platforms have developed to high standards, it has become apparent that the genomic weighting can be increased. One reason this makes sense is that pedigree is often verified through genomic testing, meaning there is a high level of consistency between the two sources. So recent testing conducted by our team at AGBU has shown that the weighting can now be increased to 95%. This testing involves a validation procedure where we first run the full TACE analysis to generate full EBVs. We then do a second analysis where we remove the phenotype records from recently born animals, and by retaining their pedigrees and genotypes, we can get a part EBV for recent animals, which we can then compare to their full EBVs. The question then is, how well do EBVs from the part analysis predict EBVs from the full analysis? And the answer is very well indeed, as we can see in this figure, which shows the range in validation accuracies across traits for the pre-genomic era pedigree model and single-step models with weightings of 50 and 95%. As you can see, there is substantial improvement in validation accuracy in moving from pedigree to single-step, and further gains by moving the emphasis on genomic information to 95%. Breeders will see some changes in EBVs with the update, particularly for genotyped animals who are not recorded for a specific trait. And as this figure shows, there will be increases in published EBV accuracies for genotyped animals, shown in green in this figure, compared to current accuracies shown in orange. So the key points to remember are that from December 2023, the weighting on genomic information will increase in the main TACE analysis from 50 to 95%. There will be some changes in EBVs, but our validations show that the result is the increased accuracy of EBVs, with the bottom line being that increased accuracy creates the potential for increased genetic gain.